Dear students, hello and assalamu alaikum. I am Ali Abbas Qazilbash and I would like to welcome you back to our program and this discussion on the topic of photosynthesis. I'm sure that you all know the basic definition of photosynthesis. Photosynthesis is the process by which green plants and some bacteria use the energy from sunlight to convert carbon dioxide and water into sugar and oxygen. The sugar is then used in various metabolic activities to produce adenosine triphosphate, ATP, the chemical fuel used by all living things. Here, I would like to add that photosynthesis, which is the conversion of unusable sunlight energy into usable chemical energy, is associated with the action of the green pigment chlorophyll. Now, the overall reaction of photosynthesis is basically 6 molecules of carbon dioxide plus 6 molecules of water will give you 1 molecule of the sugar glucose plus 6 molecules of oxygen. Green plants, in the presence of sunlight, use 6 molecules of carbon dioxide plus 6 molecules of water to produce 1 molecule of sugar plus 6 molecules of oxygen. Now, these sugars, or carbohydrates as they're better known as, are then broken down in the mitochondria of the cell to release adenosine triphosphate, ATP. Now, students, this illustration shows the conversion from glucose to ATP. It is, in fact, a drawing to show the steps of aerobic cellular reaction, which will be explained in detail in our program of respiration. Plants, especially green plants, dominate our environment, and we should be very grateful to these plants for various services they perform for us and all living things. The primary function that green plants perform is the fixing of carbon dioxide in the presence of sunlight, the process of photosynthesis, during which they use sunlight, photo, as the source of energy to synthesize food, and also produce oxygen as a byproduct of this process. Therefore, it is without doubt that photosynthesis is absolutely essential for the continuance of life because it is the source of food, both directly and indirectly, for herbivores and carnivores respectively, and oxygen for all living things. With our discussion so far, we have come to know the importance of sunlight, the radiant energy. We also know that the food produced through photosynthesis eventually becomes the food for the rest of the world. For example, humans and all other animals either eat plants directly or eat animals that have eaten plants. In other words, we can say that the living organism that produce their own food are known as autotrophs, and those organisms that do not produce their food but depend on others for their food are known as heterotrophs. Therefore, when analyzing the food chain, we can say that autotrophs are represented by green plants, algae, and a few types of bacteria, and herbivores are those animals that feed directly on plants or plant products such as cows, goats, and rabbits, whereas carnivores are those animals like snakes, lions, dogs, and cats that feed on herbivores or other carnivores. And omnivores are those animals which feed on herbivores, carnivores, plants, or plant products like humans and bears, etc. Now let us discuss the process of photosynthesis in some detail, starting with how the raw materials are supplied and used, and where the process of photosynthesis actually takes place, the chloroplast, its structure and role. Now, plants get their water, the source of hydrogen, from the soil and absorb it through their roots. Carbon dioxide reaches to the inside of the leaf, the site of photosynthesis, from the atmosphere. Carbon dioxide enters the plants through pores called stomata, present on the surface of the leaf. The opening and closing of stomata depends upon the presence and absence of light and is powered through the process of osmoregulation, the absorption of water by guard cells surrounding the stoma. The source of energy used to drive this process is, of course, radiation, sunlight, that is incident on the leaves and absorbed by specific pigments present in the chloroplast. This diagram very clearly shows a leaf and its anatomy. The mesophyll cell, with its detailed internal structure, the granum, which are stacked with thylakoids, the site where sunlight is trapped and the process of photosynthesis is initiated. At this stage, my friends, you will certainly not be surprised to know that radiation energy from the sunlight or otherwise is part of the electromagnetic spectrum, which consists of various types of radiant energies, from the shortest wavelength gamma rays to the longest wavelength radio waves. The shorter the wavelength, the greater the energy. In this diagram, you can see the different types of radiant energies. White or visible light is only a small portion of the electromagnetic spectrum, which has various wavelengths of light. In this illustration, here you can see that only violet, blue, and red lights are better absorbed by both chlorophyll A and chlorophyll B, the pigments associated with photosynthesis. Let us now discuss the structure and function of chloroplasts. The chloroplast is the site of photosynthesis. It is an organelle found in the cells of green plants and certain algae. In the chloroplast, a double membrane surrounds a large central space called the stroma. A membrane system within the stroma forms the grana, so called because it looks like piles of seeds, and early scientists, the word grana meant seeds. Now, electron microscopy later revealed that each granum is actually a stack of flattened sacs or discs called thylakoids. The grana are connected to one another by stroma lamellae. Here, you should keep in mind that chlorophyll and the yellow-orange pigments, which are called carotenoids, are found within the membrane of the thylakoids, and these pigments are able to absorb the energy of sun. The space between the chloroplasts is called the stroma, which consists of an enzyme-rich solution. These enzymes incorporate carbon dioxide into organic molecules. Therefore, we can say that carbohydrates produced by photosynthesis are formed within the stroma. Now, there are two distinct phases of photosynthesis. The first set of reactions, which takes place in the thylakoid, is called the light-dependent reactions, or simply light reactions. The second set of reactions, which takes place in the stroma, are called light-independent reactions, or dark reactions, because they do not utilize solar energy. Now we shall discuss the light-dependent reactions in some detail. The components of a photosystem are several hundred chlorophylls and carotenoid molecules, plus the reaction center chlorophyll A molecule. Now, light energy is absorbed in the form of photons and then passed from molecule to molecules, as illustrated here in yellow, until it reaches the reaction center chlorophyll A molecule. Thereafter, energized electrons are passed to an acceptor molecule. During this process, the chlorophyll is activated by a higher energy, so that its two electrons move to a higher energy state and are captured by an electron acceptor called plastoquinone. The chlorophyll replaces its lost electrons by disrupting a water molecule, and this disruption causes the water molecules to break down into hydrogen and oxygen, a process called photolysis of water. This entire process takes place within the system designated as photochemical system 2. In photochemical system 2, from the electron acceptor plastoquinone, the electrons are passed through an electron transport system. This chain consists of a series of electron acceptors, which are cytochrome B, cytochrome F, and plastocyanine. Each electron acceptor is at a lower energy level than its predecessor. This way, the electron goes on losing their energy. Some of this energy is used to make ATP from ADP between cytochrome B and F molecules. In this way, ATP synthesis is generated by the light energy and is called photophosphorylation. Similarly, electrons are passed along another pigment system at the bottom of photochemical system 1, where light energy again boosts an electron to another electron acceptor called ferredoxin-reducing substance, as you can see in this diagram. From this acceptor, the electrons are passed along to a nucleotide of adenine called NADP. It is in its oxidized form and then is reduced to NADPH. The next important set of reactions are the light-independent or dark reactions. At this stage of photosynthesis, the NADPH and ATP molecules produced as a result of light reaction are used in the reduction of carbon dioxide. The reduction of carbon dioxide occurs in the stroma of the chloroplast by means of a series of reactions known as the Calvin cycle. The series of reactions involved in the Calvin cycle include carbon dioxide fixation, carbon dioxide reduction, regeneration of ribulose bisphosphate, RUBP. The diagram illustrates all three steps. Here you can see the Calvin cycle in its simplified form. You can see that ribulose bisphosphate, which in this diagram is written as RUBP, combines with carbon dioxide, forming a 6-carbon molecule, which immediately breaks down into two 3-carbon molecules, phosphoglycerate, PGA. PGA is then reduced to phosphoglyceraldehyde, PGAL. The NADPH and ATP are used in the reduction of phosphoglycerate to a triose or 3-carbon compound termed as phosphoglyceraldehyde. This is the end product of the cycle. Outside the cycle, the two PGA molecules can combine to give glucose 6-phosphate. For every 3 turns of this cycle, 5 molecules of PGAL are used to reform 3 molecules of RUBP. Now we know that the food does not just mean glucose or carbohydrates. There are other types of food as well, like proteins, lipids, etc. And you can see that there is a net gain of 1 PGAL molecule in the Calvin cycle. And this PGAL molecule is used to produce glucose and numerous other organic molecules. From these PGAL molecules, starch, cellulose, other carbohydrates, proteins, glycerol, fatty acid molecules are produced as seen here. Let us now summarize the process of photosynthesis with the help of a few diagrams. Photosynthesis basically occurs in the chloroplasts, which are specialized organelles present in all green plants and some algae. They contain stacks of thylakoids within the grana, where the light reaction of photosynthesis occurs. In other words, solar energy is trapped by specific pigments in these thylakoids, and the water molecule is broken down into hydrogen and oxygen. This process is part of photochemical system 2. The solar radiation arrives at the reaction center where the chlorophyll A molecule absorbs the energy and releases an electron, which in turn is passed on to an electron acceptor known as plastoquinone. From here, the electron is then passed along an electron transport system from one acceptor to the next, each having a lower energy state than its predecessor. This results in the release of energy, some of which is utilized to form ATP from ADP, photophosphorylation. Simultaneously, the water molecule breaks up into hydrogen, used in the reduction of carbon dioxide, and oxygen, which is released as a byproduct. Similarly, electrons are also passed along another pigment system called photochemical system 1, where light energy again boosts an electron from the reaction center to a ferredoxin-reducing substance, which subsequently releases it to a nucleotide of adenine, NADP, which in turn is reduced to NADPH, which is used as the source of energy along with ATP to drive the dark reaction. Chloroplasts also have a large central space called the stroma. It is here in the stroma where the next step of photosynthesis occurs, the dark reaction. During this process, energy from the sunlight is not utilized. Instead, energy from ATP and NADPH molecules is used. In the dark reaction, carbon dioxide is fixed through the Calvin cycle to form organic molecules such as phosphoglyceraldehyde, which is then used to manufacture organic molecules used as food source. During the Calvin cycle, carbon dioxide is fixed through the reduction by combination with a 5-carbon molecule known as RUBP, ribulose bisphosphate. Upon combination with carbon dioxide, RUBP forms a 6-carbon molecule, which immediately breaks down into 2 3-carbon molecules called phosphoglycerate, which are then reduced to phosphoglyceraldehyde, PGAL. From these PGAL molecules, starch, cellulose, proteins, fatty acid molecules, etc. are produced. To conclude, dear students, we can say that photosynthesis is a 2-phase process by which plants manufacture their own food. The first phase consists of the light reaction, which takes place in the thylakoids, where energy in the form of sunlight is trapped by specific pigments, and some of this energy is used in the breakdown of a water molecule into hydrogen and oxygen, which is released, and the remaining energy is used in the conversion to chemical energy in the form of ATP and NADPH. The second phase takes place in the stroma and is known as the dark reaction, where this chemical energy is used to fix and reduce carbon dioxide to form a 3-carbon molecule, PGAL, which is then used to make sugar and other food substances. With this, dear students, we've come to the end of today's program. I would like to thank you all for your undivided attention. Until we meet again, wish you the very best. Khuda Hafiz.